All right, guys, let's look at number 19 here. And this is a schematic diagram of photosynthesis. So we're going to first label the light reactions right here. And then we'll go ahead and label the second part here, which is the synthesis reactions. All right, and most obviously the light reactions involve light. So here's the sun right here, we'll label that. And the synthesis reactions, the thing that is being synthesized here is glucose. All right, now let's go into what these molecules are here. And so this, fairly obviously, is water. And so when light hits the pigments on the plant's leaf, the pigments are able to absorb that energy and split water, and so what's coming off here is oxygen. In the process, hydrogens and ATP are going to be brought over to the synthesis reactions. So the hydrogens combine with carbon dioxide, and those are going to make the glucose. So if we were going to relabel this bottom part, we have the light, water, carbon dioxide, and that's going to make oxygen and glucose. All right, let's move on to another big process that we learned about. So this is cellular respiration, and it happens in 3 parts. So this should look familiar. So part A is the glycolysis, and B is citric acid cycle, and C down here is the electron transport chain. Okay, now since this is glycolysis right here, we have to think about what goes into that. And so K is glucose. All right, and what's going to be made in glycolysis is 2 ATP, and so ATP you can see is these D, E, and F labels here. All right, carbon dioxide, that is going to be produced right here. So in Krebs cycle, or sorry, citric acid cycle, CO2 is going to come off, so that's part of glucose. And so that's letter H here. Okay, NADH, these are our electron carriers, and so NAD is going to go over and pick up hydrogens and turn into NADH. And so that's going to happen at both of these steps in glycolysis as well as Krebs cycle, so I and J. All right, and those electrons with their partnering hydrogen are going to be taken to the electron transport chain. One thing we forgot down here is pyruvate. That's produced in glycolysis too, so G is there. Okay, so most of the ATP is made down here in the electron transport chain. That is around 32 to 34. Everything else is just 2, so most of it's made down here. Okay, let's keep going here. Okay, now you're supposed to label this diagram here that shows how energy changes during the course of a reaction. And the red line right here is with the enzyme. So this, we'll just mark it over here. So red is with enzyme, black is without. Okay, since our starting materials have more energy than our products, there's going to be an energy release here. And so the energy released by this chemical reaction is labeled in C. So that doesn't change. Either way, enzyme, no enzyme, that the free energy change is the same. But the only thing different is that enzymes lower the activation energy. So B is the activation energy with an enzyme. Okay, there's quite a bigger hump to go over if you don't have an enzyme. All right, let's keep going here. So enzymes are made of proteins. And what do? You can write that they control chemical reactions. Control and, let's say, catalyze, which basically means to speed up. Okay, now in our answer we have to use active site and substrate. So they control chemical reactions by taking taking in substrates into their active site. And basically within that active site, they create a favorable environment for the chemical reaction to take place. Okay, enzymes don't work in boiling temperatures because that will cause them to denature. They don't work in cold temperatures very well because the molecules are moving too slow, so they just slow down in cold temps. Okay, extreme pHs also denature all proteins, including enzymes. All right. Look at number 23. So we're supposed to label this. So our labels are down here. A nucleotide would be a base, sugar, and phosphate. So I'm just going to circle that and call that a nucleotide. Okay, deoxyribose would be one of these sugars. All right, let me just get a look at what all our other labels are. Okay. So one of these guys, okay, I'm just going to label one of these. This is a nitrogenous base. This one's cytosine, guanine, thymine, and adenine. So there's 4 different types in DNA. Our phosphate is right here. And I think we've got everything. Okay, let's look at DNA replication. So we are trying to make the complementary strand. So that's going to be A, T, G, A, G, T, 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 T. And DNA replication is the process where we make an identical copy of DNA using the original one as the template strand. So that's supposed to be in your own words, so I'll let you guys formulate that. All right. So why do we actually have to make a copy of DNA? Well, we don't have just one cell. We are going to have to make more cells all throughout our lifetime. And every time a cell splits into 2, more DNA is going to have to be made. Okay? And so every time, you guys might remember this from grammar school, this is called mitosis. But we'll just write to replace cells or grow. We need new copies of DNA. Okay, we don't actually have to do this one. It was a mistake. It's not going to be on your final, so if you found out the name of the enzyme, great, but don't worry about it for the final. But basically, you just have to know that DNA unzips, and new DNA is going to come on and attach. So if you explain that, that's awesome. All right, and let's keep going to this next part. So we're going to explain DNA technology and DNA profiling. And so we're going to, this DNA sample here is extracted DNA. So just like we did with the strawberry, you want to explain DNA extraction is where we remove DNA from the rest of the cell. You could have also written that we're going to do PCR to make sure that we only have the STR regions for analysis. Okay, and then we're going to introduce restriction enzymes, which are going to cut the DNA, and those are going to go in our wells. And DNA is negative, so it travels towards the positive end of the electrophoresis machine. So this is gel electrophoresis. All right. And then the electricity is going to separate out the fragments by size, and we'll be able to compare the crime scene with all the suspects. Okay, let's take a look. So how many bases is the uncut DNA? So that's right here, and they tell you the size on this side. So this is probably 6 bases. All right, so digested DNA means that it was cut with restriction enzymes. How many times was the DNA cut to produce the results seen to the left? So this is digested DNA here. You see that it's 2 pieces of DNA. So the 2 pieces must have come from just one cut. All right, and the smallest fragment down here, you can see it's a little bit bigger than 2, but not quite 3. So I'm going to say about 2.2 is a good estimate there. Okay, for number 28, so let's see here. So we're going to have to draw in the other people's bands. So you could see the 550 is probably this one right here because it traveled further because it's smaller. And this is probably our 900. So I'm just going to mark these so we remember. And so this one's going to have a piece at 50 and 1200. So 50 is super small. That probably went really far. 1200 is bigger than 900, so that's probably going to be up here. And this one's got a 100 and a 250, so 100 is probably pretty close to our 50 over here, so that's going to be about right there. And the 250, oops, disappeared, is going to be about right there. Okay, so you just estimate based on this center, these center bands, to figure that out. All right, in number 29, we need to actually explain PCR. So go through each of these steps and talk about what happened. So in step 1, you just had to say that this is the sample DNA. And in step 2, we're going to heat it to over 90 degrees Celsius. And that's going to unzip the DNA or separate it. So it's going to break the hydrogen bonds between the 2 sides of the DNA. And those are going to come apart. And we also, into our PCR tube, need to add primers. So primers kind of guide the enzymes as to where to start copying the DNA. Okay, so primers attach at a cooler temperature. Okay, and then our enzymes, specifically a certain enzyme called DNA polymerase, is going to go through and put new nucleotides down. Okay, so then new nucleotides are added. And then we are going to get 2 identical pieces. And number 5, this process is just going to repeat again. We're going to heat up the DNA again. More primers are going to attach. New nucleotides are going to come in. And all of a sudden, we're going to get tons and tons of DNA copies. All right, and that's the end of our review.